Hello dear students, today we are going to study one more chromatographic technique is paper chromatography. In last video we studied thin layer chromatography. So here thin layer chromatography and paper chromatography both are same. The only difference is in thin layer chromatography we use glass plate which is coated by a silica and alumina gel as a stationary phase. Here instead of that, here we use chromatographic paper. Remaining each and everything is same here. In paper chromatography is a technique of separation and identification of chemicals by solvents, means mobile phase, diffusing through a strips of filter paper on which spot containing mixture of components is placed. So paper chromatography is same as thin layer chromatography. This is a closed container which prevent the evaporation of mobile phase or volatile solvent, and this is a chromatographic paper. And here we place the sample mixture here 1 centimeter from the base, and when the mobile phase starts to move upward direction, the components of mixture also starts to move. So this is paper chromatography. So here these are the different bands, means these are the different components. So in paper chromatography, depending upon the solvent is moving upward or downward, this is also classified into two types. One is ascending chromatography, one is descending chromatography. So if our mobile phase starts to move upward direction, that is ascending chromatography. In case if mobile phase is starts to move in a downward direction, that is descending chromatography. So these are the classification of paper chromatography, or else the procedure is same. Here what happened, when we a small spot of a sample which is placed about 1 centimeter from the base. When a paper is dipped in a mobile phase, the mobile phase starts to move in the upward direction and it carries a sample. And these are the different components or different bands. And finally components present in the sample get separated into various bands. So from that we can calculate the components which are present in a sample mixture. And next chromatographic technique is column chromatography. Here this is called column. This is column chromatography. In this technique, a solid stationary phase. Here in some column chromatography, stationary phase is used in a as solid is an adsorbent material such as same silica gel and alumina gel packed in a column. So this is stationary phase. This entire is column. So here what happened in this technique, this solid stationary phase, this is a solid stationary phase is an adsorbent material such as silica, alumina, which is packed in a column, and a glass tube with a stop cock at the lower end is taken, and a small plug of cotton is placed here, which is prevent the movement of stationary phase through this cock. And here what happened, when a concentrated solution of a given sample mixture is starts to pour here, when we load this sample solution at the top of the column, as the solvent flows down, as the solvent starts to flow down through the top layer, it dissolves the mixture, and as the solution runs through the adsorbent, the components of mixture are subjected to a competition between two phases. The stationary phase tries to absorb the components on its surface. Here the solid stationary phase means silica or alumina, which tries to attract the components which are present in a loaded sample. So here its surface, while mobile phase tries to take away the components along it downwards. As a result, we get different components are adsorbed at a different height here from the bands. And here we mentioned weaker interaction means this component is weak to interact with the stationary phase, and these components are move along with mobile phase easily. We can collect it here, and this mobile phase, and this one is a strong interaction which interact with the stationary phase, and it will take much time to move along with this column. This is column chromatography. And one more topic is there is solvent extraction. Solvent extraction means what? And an organic compound. Usually organic compounds are dissolved in organic solvent, but some of organic compounds also soluble in water and present in the aqueous solution. So if we need to take out from the organic compounds from aqueous solution, so we have to use the organic solvent in which the compound is more soluble compared to water. So definition of solvent extraction is when a solute is added to a mixture of two immiscible liquids, the solute itself gets distributed between the two liquids. One is two liquid means one is aqueous media, another one is organic medium. So in such a way that the ratio of concentration of solute in organic layer is to concentration of solute in aqueous layer is constant at a constant temperature. This is the definition of solvent extraction. So here what happened? Here the mixed aqueous solution of organic compounds. Here this is a aqueous medium, and these red colors are the organic compound. So these organic compounds are mixed in the aqueous phase. So when we add the organic solvent, suitable organic solvent, and this is done by a separating funnel. So when you shake this funnel, what happens? All the organic compounds present in the aqueous phase are starts move towards the organic compound, because we already said the organic solvent in which the compound is more soluble compared to water. So from this technique we can extract the compound from aqueous solution. This is nothing but solvent extraction.